Young anatomists, grab your colors and let's get drawing. This video is going to talk about neuron structure and function. A neuron is the key functional unit, a cell type that is in the nervous system. And so let's start by drawing a neuron and talking about the basic parts of a neuron. So here's a neuron cell. Neurons have a very interesting shape. And the shape of a neuron and the structures of a neuron really fit the functions very well. You can see that the ends of the neurons have very distinct and different shapes and structures. This end of a neuron down here has a lot of structures called dendrites. And this doesn't really have any dendrites, so I'm going to draw them in. So dendrites are these branched structures that come off of one end of the neuron. And you can see that there's a bunch of branches here. So dendrites are highly branched. And dendrites receive inputs. So by having all of those branches, this neuron can touch a bunch of different cells and receive all of those inputs at the same time. Just like most cells, neurons have a nucleus, so there's our little nucleus, and this portion of the cell is called the cell body. Cell body. And sometimes it's called the soma. And then this part of the neuron is really cool as well. This part is called the axon. And the axon kind of has the opposite function of the dendrites. Whereas the dendrites receive inputs, the axon sends messages, has outputs. And the axons are highly branched as well. And this way, one single neuron can send output messages to a bunch of different cells. So the signal that passes through a neuron goes this way, from the dendrites into the cell body, and then to the axons. So let's draw some signals coming in. So this dendrite could be touching a cell and getting a signal and sending that signal to the neuron. This dendrite could be touching another cell, send a signal in. So neurons are getting all of this information from a bunch of different cells at the same time. That information is integrated in the cell body, and then that information is sent down the axon and can be sent to a bunch of different cells simultaneously. So this part down here is called an axon terminal, and terminal just means end, right? So this is just saying that's the end of the axon, and there's some special stuff going on over there that we'll talk about in just a second. And each one of these axon terminals is going to touch another cell. So let's say that this neuron was communicating with another neuron. Well, then that neuron would be right there. There's its cell body. There'd be dendrites coming out of all of these places. And then this neuron would have an axon that would send branches to the next cell that it communicates with. And so this is how neurons send signals to each other and how they communicate. All right, I want to tell you about two parts of the signal in a neuron. Neurons use electrochemical signaling. And what that means is that there's an electric part to the signal and there's a chemical part to the signal. And that's what we're going to mostly be dealing with on the rest of this page. We're going to treat the electric part separately from the chemical part. So let's dive into that electrical part. Actually, there's something else that I want to tell you before we get off of this part. So a neuron, a lot of people confuse these words, a neuron versus a nerve. So a neuron is a single cell, and a nerve is just like a bundle of neurons. All right, so let's dive into this electric part of the signal. And where we're going to focus in on this is we are going to focus right in on here, and we're going to draw a section of that cell membrane right down here at the bottom. So this part of a neuron's function is called an action potential. And in a neuron's signal, the action potential is the electric impulse. All right, so let's draw in this membrane here. So here's our neuron cell membrane. So let's write that. This is our axon membrane. And we're gonna make this go into 4 different stages. So here's stage 1 of this process, stage 2, stage 3, and stage 4. Now the main players in sending this electric impulse are drawn in right here. So let's color these in. So we've got this guy right here, and the name of this protein is called the sodium-potassium pump. And the job of the sodium-potassium pump is to pump sodium out and bring potassium in. And the sodium-potassium pump uses a lot of ATP to do this. Uses ATP. Now, the other main players of this process are some ion channels. Ion channels. And these ion channels also have to do with sodium and potassium. So we'll draw them as some basic shapes here. So this shape is our potassium channel. And this shape right here is our sodium channel. So in step 1, the sodium-potassium pump is hard at work bringing all of those sodiums outside of the neuron and bringing all of those potassiums inside of the neuron. So I guess we should set that up too. So this is the outside of the cell, and this part right here is the cytoplasm, which is the inside of the cell. So we're bringing all of this potassium in and pushing all of that sodium out. The reason why these molecules are very important is because they're ions. Ions are charged atoms. And that's why this signal is able to be electric, because we're using charged atoms. Now, some people to remember which is on the outside and which is on the inside of sodium and potassium, there's a mnemonic called salty banana. Banana. So what that means is if you imagine a banana, right? And when you think about bananas, you think about bananas being like high in potassium. And if you imagine a banana that's salty, salty means sodium. So the banana has potassium on the inside. Salty banana has potassium high on the inside and sodium high on the outside. Salty banana. All right, so our first step of things has the sodium-potassium pump working to pump sodium out and potassium in. Let's see what's going on in step 2. So we've got the same players. Here's our sodium-potassium pump. And our sodium channel and our potassium channel. But during step 2, the sodium channel is very active in bringing sodium in. So now we're increasing the amount of sodium on the inside. Step 3, we're thinking about the same players here, but the one that's active is going to be different. It's going to be our potassium channel that's sending potassium out. And then step 4, those ion channels calm down. And it's back to work for the sodium-potassium pump. So now we're back to potassium being pumped in and sodium being pumped out. So when we say that neurons use electric signals, what ends up happening is we're just changing the location of these ions, of these charged atoms. And you can kind of like track that change in charge by using a graph like this. Let's draw that in. This graph shows the change in electrical activity across the membrane of a neuron during an action potential, during an electric signal. And so what we have on our x-axis is time, and the amount of time we're talking about is in milliseconds. So neuron signaling happens really fast. And then along the y-axis, what we're dealing with is voltage. And our units for that are millivolts. Now, one important number for us to remember is this one right here. And this is minus 70 millivolts. And during stage 1, when the sodium-potassium pump is working really hard, your neuron is at rest, and minus 70 millivolts is called our resting membrane potential. So let's write what's going on up here, down at the bottom. So at this point, potassium is coming in, and sodium is going out to bring our voltage to minus 70. And then what happens when the neuron gets the signal, you can see that there's a change in the voltage. And this voltage goes all the way to somewhere in the neighborhood of about plus 40 millivolts. And the trigger for this is a stimulus. Can be a chemical stimulus, can be a temperature stimulus, pain, something like that. But this next stage of the action potential we call depolarization. And during depolarization, sodium is the mover. So sodium moves in. And hopefully you can imagine why this charge would be increasing. Sodium is a positive charge. If that positive charge is coming into the cell, that's going to cause the voltage to increase. And then what happens is that increase in voltage gets to a certain point, and it triggers those sodium channels to close and the potassium channels to open, and we get to stage 3 on here. Stage 3 of an action potential is called repolarization. In repolarization, what's happening is that the potassium is now moving out. Potassium is also a charged ion, positively charged ion. So when all of those positive charges are flooding out of the cell, that's what causes the voltage of the cell to go down. What ends up happening is that voltage overshoots the resting potential, and that overshoot is what triggers these potassium channels to close. This part of things down here is called hyperpolarization. And at hyperpolarization, these doors shut and your sodium-potassium channels kick in again to pump sodium out and potassium in, reestablishing our salty banana and reestablishing our membrane potential back to rest so that your neuron can charge back up and fire an action potential again. So this part is the part that's the electrical signal. Now, the other part of things is the chemical signal, and we're going to draw that up here. So our chemical signal is called synaptic transmission. And synaptic transmission happens at something called a synapse. So let's draw a synapse. What exactly are we drawing? Well, we're zooming in right to this area, the area between the axon of one neuron and the dendrite of another. So this is what we're zooming in on right here. And this area is called a synapse. Well, let me draw in the cells first, I guess. So here's our axon of one neuron. And let's use another color for the dendrite of our next neuron. Oh, I didn't really leave myself a lot of space. All right, so dendrite of our next neuron there. And we call these neurons, we call this one the, I use that same color, we call this one the presynaptic cell. And we call this one, then hopefully you kind of got it, the postsynaptic cell. And if this is presynaptic cell and this is the postsynaptic cell, what we call this part in the middle is the synaptic cleft. A synaptic cleft, it's the gap. So it's the tip of one neuron, then a gap, and then the tip of the next neuron. And together, these three structures, the presynaptic cell axon, the synaptic cleft, and the postsynaptic cell dendrite, those three structures together are called the synapse. And so we're going to talk about what happens with a chemical signal that passes from one to the other. A critical structure involved in this is in the presynaptic cell axon, this axon terminal. We've got some interesting structures that are bubbles or containers called synaptic vesicles and synaptic vesicles contain the chemicals of neuron signaling. So these chemicals are called neurotransmitters. So neurotransmitters are the chemical signaling molecules. And that's going to send a chemical signal across to the dendrite. Now, one cool thing about the dendrite on the postsynaptic cell is that there are receptors on there. And these receptors, they help the dendrite receive the signal. So these, let's label it right up here. These are receptors, and they're specific for neurotransmitters. So I'm just going to put NT for neurotransmitters. All right, so we got that. A synapse is made up of a presynaptic cell axon, a postsynaptic cell dendrite with a gap called the synaptic cleft in the middle. We're going to use neurotransmitters to do chemical signaling from one neuron to the next, and this next neuron's dendrites are able to receive that signal because they have specialized neurotransmitter receptors. All right, let's talk about this process in a little bit more detail about what happens. So this neuron would actually be what's happening at rest. Everything is ready to go up here. And down here, we're going to draw and outline the steps of synaptic transmission, that chemical signal part. All right, so that's what's going on down here. So let's draw it in again. As you're drawing, try to practice and say some of these terms. So this is the presynaptic axon terminal, our postsynaptic dendrite, our dendrite on our postsynaptic cell has neurotransmitter receptors that are really specialized. Our presynaptic cell has these beautiful synaptic vesicles, and those synaptic vesicles are filled up with neurotransmitter chemicals. All right, so what happens here? Well, step one of synaptic transmission is that the action potential has to come down this axon and get to the end. So let's write that in. Step one is the action potential comes to the end of the axon. And once this action potential gets to the end, that action potential is going to kind of like put a buzz here in these synaptic vesicles. So that's step two, is what happens is something called exocytosis. And we'll talk about what that means in just a second. Exocytosis of neurotransmitters. So these synaptic vesicles, they move to the very end of the axon terminal, and they end up fusing with the end. They bind to, they merge with the end, and then the chemicals that were in there, those acetylcholine or those neurotransmitter chemicals, acetylcholine is one type, those neurotransmitters spill out into the synaptic cleft, and that's the part that's exocytosis, where the synaptic vesicle goes to the end of the cell, spills out its contents outside of the cell, and now these neurotransmitters can bind to the postsynaptic cell. So let's write that in as step three. Step three is that the neurotransmitters bind receptors. And then this chemical signal causes a response in the postsynaptic cell. Well, what is that response? What happens? And the truth is that it kind of depends. So let me tell you what I mean down here. What do I mean that this response depends? Well, let's draw another graph down here, and this graph is going to be a copy of the graph on the other side, where we have our time and we have our voltage. And now we're measuring that voltage of the postsynaptic cell. Now, our magic number here, we said, was minus 70 millivolts. And so that postsynaptic cell is just chilling at minus 70 millivolts at rest. And one of two things can happen over here with the response. And that depends on which type of neurotransmitters are being used here. We can have neurotransmitters that are excitatory, or we can have neurotransmitters that are inhibitory. And excitatory neurotransmitters cause a signal to happen. And inhibitory, hopefully you see that word in there. So they shut it down, no signal. They shut down the signal. And how does that happen? Well, what happens is if an excitatory neurotransmitter is used, that increases the voltage of the postsynaptic cell. And once that voltage reaches this level right here of minus 55 millivolts, then you get the whole action potential happen. And once this minus 55 millivolts is reached, that's kind of the magic number to cause this response. And this is called the threshold potential. So once minus 55 is reached, this whole response happens. Let's draw that in over here too. What's interesting about how neurons work is once this threshold is released, you get a maximum action potential. The whole thing happens, and this is called the all or none principle. All or none. So the action potential, or the neuron, fires completely, all, or not at all, none. And so when a neuron sends a signal, it's all in, right? It only knows kind of like one strength of signal. So if that's true, and neurons are in charge of sensations like pain, how do you get pain at different levels? Well, it's just the number of signals that are sent there, rather than the signal being like a stronger peak or anything like that. All right, so this excitatory neurotransmitter causes the postsynaptic cell to reach its threshold potential. But if we have an inhibitory neurotransmitter, the inhibitory neurotransmitter sends the voltage of the postsynaptic cell down, and if that threshold potential is never reached, then there's never going to be a signal in the postsynaptic cell. It just shuts that signal down. So that's how you can have two different flavors of neurotransmitter causing different responses in the postsynaptic cell. Now, there's one other thing that I want to tell you about in this final little slice of our notes page right here. And that is a special type of signaling in a neuron. And that special type of signaling is called saltatory conduction. And saltatory conduction gets its name from the same roots as the Spanish word saltar, which means to jump or to leap. And the type of signaling that we've talked about so far is just kind of like our regular signaling. And neurons are built for speed. One of the ways that neurons are built for speed is by having those neurotransmitters all pre-packaged and ready to go, so that when a neuron receives the go signal, all it has to do is slightly move those neurotransmitters and not make them from scratch. Another thing is that electrical signal, those ions are ready to go, and it's kind of like how a mousetrap is set, and that mousetrap is ready to go. As soon as a mouse steps on it, that mousetrap can snap. So normally a neuron sends a signal from its dendrites, soma, into the axons, and a normal signal goes at about 11 miles per hour, which doesn't seem that fast, but when you consider the length of your body, that's really, really fast. But neurons can make some changes when it's important that a signal goes faster. So let's draw another neuron down here and think about these parts as you're drawing them. So I'm drawing some basic branched dendrites over here. We have our cell body with a nucleus, our long axon with our axon terminals at the end. And right now, it looks pretty much the same as the picture above. But what we're going to add is we're going to add these superhero cells up here on the axon. So I'm going to draw those in. And each of these things right here are called Schwann cells. And it's named after a person, Dr. Schwann, that kind of discovered those cells or described those cells. And Schwann cells provide a substance to the axon called myelin. And myelin acts like insulation. So just like you have insulation or like plastic or rubber on the outside of a wire, you can have myelin insulation that protects the electric signal on the axon. There are gaps in between the Schwann cells, and those are called nodes. And more specifically, they're called nodes of Ranvier. And that's another thing named after a person. So up here, this type of axon that's, kind of like the normal one, we would call it non-myelinated. And down here, the one that's built for speed is a myelinated neuron. And what happens is that instead of the signal having to pass through every bit of the axon, that signal jumps from one node to the next to the next. So the signal gets to skip a lot of that distance. And how this helps is that this signal ends up going about 330 miles per hour, about 30 times faster. So myelinated neurons are built for speed. How are they built for speed? Because those neurons have myelin, which is insulation to protect and preserve a strong electrical signal in the axon. All right, so what have we done here today with these drawing notes? These drawing notes are meant to feature the neuron, the main functional unit of a cell. A neuron works and sends signals to other neurons using electrochemical signals. The electrical part of the signal is called an action potential. An action potential involves moving sodium out first and moving potassium in to create this neuron that's like a salty banana with potassium high on the inside and sodium high on the outside. When a neuron gets a stimulus, gets a signal, where these ions are change shape. First, sodium comes in. Next, potassium goes out. And that's what causes this electrical signal, because these sodium and potassium are ions, charged atoms. And that causes this change in voltage along a neuron. Once that action potential reaches the end of a neuron, the axon terminal, that causes the chemical signal, which is synaptic transmission. Synaptic transmission involves exocytosis of the neurotransmitters, spilling those neurotransmitters out into the synaptic cleft, the gap between the two signaling cells. And then the chemical signal goes to the postsynaptic neuron, where neurotransmitters bind to neurotransmitter receptors. Those neurotransmitters can be excitatory and cause a signal in the postsynaptic cell, or inhibitory and stop a signal from happening in the postsynaptic cell. So hopefully this gets a lot of information all on one piece of paper and makes your journey through the nervous system that much easier.